So we just talked about the factors preventing collapsing tendency of lungs. That include two main factors: intrapleural pressure, preventing collapse by elasticity, and the surfactant, preventing collapse by surface tension. Now we are going to discuss them in detail here. Let me start with the surfactant first. Well, surfactant is a surface-acting material or agent that is responsible for lowering the surface tension of fluid. When it lines the epithelium of the alveoli in lungs, is known as pulmonary surfactant and decreases the surface tension on the alveolar membrane. Now, where does this secretion of pulmonary surfactant come from? Now, pulmonary surfactant is secreted by two types of cells. Type 2 alveolar epithelial cells in the lungs, which are called surfactant-secreting alveolar cells, or pneumocytes. Characteristic feature of these cells is the presence of microvilli on their alveolar surface, while the other one being the club cells or Clara cells, which are situated in the bronchioles. These are shown here in blue. Now, if I remember from my previous video, I mentioned another name for them. Yes, they are also called bronchiolar exocrine cells. And one of the primary functions of these cells is to secrete surfactant. I mentioned in the last part of the video that one of the factors having the tendency to collapse the lungs is surface tension. Now here you can see the alveoli, and here a layer of fluid lines the alveoli. This is the reason that produces the surface tension in the alveoli. That is directed towards the center, hence trying to collapse the lung. If you recall the chemistry, there's a type of bonding called hydrogen bonding present among the water molecules. Due to the electrostatic force of attraction between negatively charged oxygen atom of water molecule and positively charged hydrogen atom of neighboring water molecule. So, a water molecule is being attracted from all sides, hence the net force being zero. While at the alveolar air fluid interface, these forces are unbalanced, because water molecules are not surrounded by other water molecules from all sides, so the net force is not zero here. Rather, these molecules on the surface will pull each other closer, hence shrink to the small surface area. This force is called surface tension. That tends to collapse the alveoli. This is a manifestation of Laplace's law, which shows that the pressure in a bubble, or alveolus, that is trying to collapse it, is directly proportional to the surface tension, T, of the molecules, and inversely proportional to the radius of the alveolus. A small bubble will therefore have a higher collapsing pressure than a larger one, and if connected, will collapse into it. Higher pressure will require higher force to overcome it in order to inflate the lung. Hence, it would be difficulty to breathe in, because inspiration requires the lungs to be inflated. The inward force created by surface tension also tends to suck fluid into the alveoli. That is called transudation. Now, luckily, we are blessed with a mixture of phospholipids secreted by type OE pneumocytes, the surfactant, that minimizes these problems. Surfactant is a lipoprotein complex formed by lipids, especially phospholipids, proteins, and ions, that are mostly calcium ions. Major phospholipid present in the surfactant is dipalmitoyl phosphatidylcholine, or DPPC. Other lipid substances of surfactant are triglycerides and phosphatidylglycerol, PG. Also, there are some proteins called specific surfactant proteins. There are four main surfactant proteins called SPA, SPB, SPC, and SPD, where SPAA and SPE are hydrophilic and have a role in producing an immune response as they help in opsonization, while SPB and SPC are hydrophobic and play active role in reducing surface tension. Now, phospholipid molecule, DPPC, in the surfactant has two portions. One portion of the molecule is hydrophilic. This portion dissolves in water and lines the alveoli. Other portion is hydrophobic, and it is directed towards the alveolar air. This surface of the phospholipid along with other portion spreads over the alveoli and reduces the surface tension. Now, during the inspiration, alveoli are inflated. This increase in the size of alveolus will obviously make the surfactants far apart from each other. Lesser concentration of surfactant then leads to the decreased effect, and the surface tension increases, thus decreasing the expansion rate of the alveolus and increase the ability to recoil. Now, during the expiration, as alveoli shrink and the fluid surface area is reduced, surfactant is thus concentrated, further reducing surface tension and preventing alveolar collapse, and helps maintain similar alveolar sizes and reduces lung stiffness and transudation. Now, surfactant plays an important role in the inflation of lungs after birth. In fetus, the secretion of surfactant begins after the 3rd month. Until birth, the lungs are solid and not expanded. Soon after birth, the first breath starts because of the stimulation of respiratory centers by hypoxia and hypercapnia. Although the respiratory movements are attempted by the infant, the lungs tend to collapse repeatedly, and the presence of surfactant in the alveoli prevents the lungs from collapsing. Absence of surfactant in infants causes collapse of lungs, and the condition is called respiratory distress syndrome or hyaline membrane disease. Not only in infants, but the deficiency of surfactant occurs in adults also, and it is called adult respiratory distress syndrome, or ARDS. In addition, the deficiency of surfactant increases the susceptibility for bacterial and viral infections. Now, moving on to the second factor preventing lung collapse, the intrapleural pressure. Well, there are two types of pressures that are exerted in the thoracic cavity and lungs during process of respiration. Intrapleural pressure or intrathoracic pressure, intraalveolar pressure or intrapulmonary pressure. Intraalveolar pressure, also known as intrapulmonary pressure, is the pressure existing in the alveoli of the lungs. Normally, intraalveolar pressure is equal to the atmospheric pressure, which is 760 mm of mercury. It becomes negative during inspiration and positive during expiration. Intrapleural pressure is also called intrathoracic pressure, since it is exerted in the whole of thoracic cavity. It is actually the pressure existing in pleural cavity, that is, in between the visceral and parietal layers of pleura, and is exerted by the suction of the fluid that lines the pleural cavity. Now, under physiological conditions, the intrapleural pressure is always negative, and is measured by direct method and indirect method. In the direct method, the intrapleural pressure is determined by introducing a needle into the pleural cavity and connecting the needle to a mercury manometer. In indirect method, intrapleural pressure is measured by introducing the esophageal balloon, which is connected to a manometer, and the intrapleural pressure is considered as equivalent to the pressure existing in the esophagus. Now, there's another type of pressure that is worth mentioning here. It's called the transpulmonary pressure, or transneural pressure. It is actually a pressure difference between the two pressures we just discussed, the intraalveolar pressure and the intrapleural pressure. You can say that it's the measure of the elastic forces in the lungs that tend to collapse the lungs at each instant of respiration, called the recoil pressure. So now you must have a better idea of how your lungs are prevented from collapsing.